Lipids 101, what they are and how we classify them. Welcome everyone. Today, we begin an in-depth exploration of lipids, one of the four essential pillars of biological macromolecules. While often reduced to simple fats in common parlance, lipids are chemically diverse and physiologically indispensable. This lecture will serve as a comprehensive foundation, bridging the gap between molecular structure and clinical health, and will explore why these molecules are the unsung heroes of cellular function and human survival. Learning objectives. By the conclusion of this session, you will achieve four primary goals. First, defining lipids through the lens of their unique physical and chemical properties. Second, mastering the tripartite classification system, simple, compound, and derived lipids, which allows us to categorize them by their chemical composition. Third, understanding the structural nuances of fatty acids, specifically how saturation influences their physical state. Fourth, connecting these biochemical principles to their massive role in human physiology and pathology, including metabolic diseases. What are lipids? The term lipid is derived from the Greek lipos, meaning fat. Scientifically, lipids are defined not by a shared structural unit, but by their solubility profile. They are a heterogeneous group of water-insoluble or hydrophobic organic compounds. Their defining trait is their inability to dissolve in water, which forces them to dissolve in nonpolar solvents like ether, chloroform, or benzene. Unlike proteins or carbohydrates, they are not polymers, which allows them to serve as unique, modular building blocks for biological membranes. General characteristics of lipids. Lipids are composed mainly of carbon, hydrogen, and oxygen, but with a significantly lower proportion of oxygen than carbohydrates. This low oxygen content is critical. It allows for a higher degree of oxidation and, consequently, a much higher energy density. While other biomolecules provide roughly 4 kilocalories per gram, lipids provide approximately 9 kilocalories per gram. Their nonpolar nature is governed by long hydrocarbon chains that repel water, effectively partitioning them away from the aqueous environment of the cell to form distinct structural barriers. Importance of lipids. Lipids are fundamentally multipurpose molecules. They serve as the body's primary long-term energy battery within adipose tissue. Structurally, they are the architects of the cell membrane, forming the flexible lipid bilayer that allows for selective permeability. They also act as chemical signaling molecules, including steroid hormones that regulate growth and metabolism. Furthermore, they are the only reason we can absorb fat-soluble vitamins A, D, E, and K. Without lipids, these vitamins would remain inaccessible to the body. Classification overview. To navigate the complexity of lipids, we divide them into three categories based on the products of their hydrolysis. Simple lipids, these are esters of fatty acids combined with various alcohols. Compound lipids, these are more complex, incorporating fatty acids and alcohols with extra chemical groups like phosphate, nitrogen, or carbohydrates. Derived lipids. These are the subunits formed after the breakdown of the first two groups, providing the essential building blocks like fatty acids and steroids. Simple lipids, fats, oils, and waxes. Simple lipids are the most basic form of stored chemical energy. Fats and oils are essentially triglycerides, esters of glycerol and 3 fatty acids. The distinction is physical. Fats are typically solid at room temperature because their structure is saturated, while oils are liquid because they are unsaturated. Waxes, while less discussed in metabolism, are essential esters of long-chain fatty acids and alcohols, providing waterproof barriers for plants and animals. Compound, complex, lipids. Compound lipids are sophisticated molecules that perform more than just storage. Phospholipids contain a phosphate group and are critical for cellular architecture. Glycolipids feature a carbohydrate group and are the primary agents of cell-cell recognition. Lipoproteins are specialized complexes that combine lipids with proteins, acting as the essential transport system to move nonpolar fats through the aqueous bloodstream. Derived lipids. When the body hydrolyzes complex lipids, it releases derived lipids. This group includes fatty acids, glycerol, and steroids like cholesterol. While they sound simple, they are the most active components of metabolism. Steroids, for instance, share a unique four-ring structure that allows them to modulate membrane fluidity and serve as the precursors to all steroid hormones. This group also encompasses the fat-soluble vitamins, which are biochemically classified here due to their lipid-dependent nature. Fatty acids, the building blocks. Fatty acids are the fundamental unit of most lipids. Structurally, they are long hydrocarbon chains capped by a carboxylic acid group, COOH. This creates an amphipathic molecule. The acid end is polar and hydrophilic, while the long tail is nonpolar and hydrophobic. This dual nature is what allows fatty acids to interact with both the watery interior of a cell and the fatty core of a membrane. Saturated versus unsaturated. The physical state of a fat is dictated by its bonds. Saturated fatty acids contain only single bonds, which allows the hydrocarbon chain to remain straight, packed together tightly, and exist as a solid at room temperature. Unsaturated fatty acids contain at least one double bond, which creates a permanent kink in the chain. These kinks prevent the molecules from packing closely, keeping them liquid and mobile at room temperature, which is essential for biological fluidity. Essential fatty acids. The human body is remarkably capable of synthesizing many things, but it lacks the enzymes to place double bonds at certain positions in fatty acids. This makes certain fatty acids, like omega-3 and omega-6, essential. They must be acquired through diet. These aren't just for energy, they are the required precursors for molecules that regulate the inflammatory response and brain function. Phospholipids, membrane architects. Phospholipids are the reason life can exist in distinct compartments. They possess a polar phosphate head and two nonpolar fatty acid tails. When placed in water, they naturally self-assemble into a bilayer with the heads facing out and the tails tucked into the center. This bilayer is the standard foundation of the plasma membrane, acting as a selective gatekeeper that regulates the internal environment of the cell. Cholesterol. Often misunderstood, cholesterol is an indispensable steroid lipid with a complex four-ring structure. It is embedded within the cell membrane to act as a fluidity buffer. At cold temperatures, it prevents the membrane from freezing into a solid, and at high temperatures, it prevents it from becoming too loose. Beyond membranes, it is the raw material for synthesizing all steroid hormones, testosterone, estrogen, etc., and bile acids required for fat digestion. Biological function summary. To tie this together, lipids are the body's multitaskers. They manage energy through high-density storage, they provide the structural scaffolds for all cells, they act as the letters in the chemical signaling system through hormones, and they provide the thermal and mechanical buffers, cushioning, that protect our vital organs. Without the versatility of lipids, the body could not maintain its internal temperature, structural integrity, or metabolic signaling. Clinical significance. When these metabolic pathways fail, clinical issues arise. Hyperlipidemia, or an excess of lipids in the blood, is a significant risk factor for cardiovascular disease. This often leads to atherosclerosis, the hardening of the arteries due to the accumulation of lipid-rich plaques. Furthermore, when lipid storage capacity is overwhelmed, it results in obesity and systemic metabolic syndrome, proving that lipid balance is central to modern medicine. Summary. We have covered a vast terrain, from the definition of lipids based on their hydrophobic solubility to the specific classification into simple, compound, and derived lipids. We have seen how the molecular geometry of a fatty acid, its saturation, polarity, and shape, dictates its ultimate role, whether it is fueling a long run, insulating an organ, or forming the very walls of our cells. Thank you. Thank you for your close attention to this foundational topic. Understanding lipid biochemistry is not just an academic exercise, it is the prerequisite for understanding pharmacology, human nutrition, and the pathology of most metabolic diseases. I am now happy to open the floor to any questions regarding the structural or functional aspects we have discussed today.